Hello. In the olden days, people knew or could identify the children based on the features. How did they look like? Do they look like mother? Do they look like father? Do they look like the grandparents? So in a way, people knew that something is being inherited by the children from the parents, and this cycle keeps on going on happening. And in that case, so there were trying to understand how did this process happen. And there were, since the time we started agriculture or animal husbandry, we know the basis of what is artificial selection. We know what kind of traits parents to take and allow the breeding among between them, so that, or crossing, so that they can get offspring with similar phenotypes or and so on. That is artificial selection. What this all means is humans have started knowing it and trying to perform these selection processes without knowing how these actually happened. And that is where science comes. And different people proposed different ideas about how these things could be happening. Say, for example, that of what was proposed by Darwin is something like different characteristics that are present from the different bodies. The body parts would come into the gametes, and from gametes they would fuse with other gametes of the female and then give rise to an offspring. And that is how he has proposed, apparently, that the characteristics are inherited by the next generation. Mendel was about the same time, but they didn't communicate much. And Mendel has worked on plants, and he did extensive work. He has multiple qualifications and expertise in different areas. He is also a monk, and he performed a lot of experiments. And one of the best things about it is how he has recorded his observations. He designed his experiments very well, recorded them very well, and pondered on that data, a large amount of data, and then came up with the laws of inheritance. So at the time when Mendel has proposed or did his experiments, and he gave two lectures, but nobody understood them or weren't taken seriously. And he has publication which did not receive as much. And similarly, he wrote to several biologists, but either they were uninterested or did not understand the findings as such. He did doubt his own work because people haven't responded well or taken up his observations. And apparently he also said something like this: "My time will come." It took a lot of time for his time to really come. I'm unsure if he was alive to see the significance of how much the importance of his work became to be. In that time, there were three scientists, almost 50 years or so, or a generation different: Hugo de Vries, Carl Correns, and Erich von Tschermak. These three botanists were also doing similar experiments, and they couldn't understand, or probably it's said that a couple of them might have understood their results better after reading Mendel's work. And then these three scientists they discovered Mendel's work. Mendel has discovered the laws of inheritance, you can say. And these people discovered Mendel's work, and that is how they have attributed or gave the credit to Mendel. That's a nice thing they did. I'm Dr. B.C.M.R., Assistant Professor at Shastra. We are going to discuss the laws of inheritance, and we will talk first about the law of segregation. Before going into that, we will discuss about the different traits that Mendel has used for his work. We are not talking about law of dominance as such, but we already discussed that, which would mean there are some traits or alleles. Some alleles exhibit their phenotype, while some alleles of the same gene could not. And those, if the presence of that allele is dominant over the phenotype of the other allele, and those are said to be as dominant. We will have a look at it. So there are seeds. Mendel has worked on several, about 7 or 8 characteristics, and he looked at the traits carefully, and some of them would be something like seed shape, round or wrinkled. And so round, the ones that are described on the top or displayed on the top are the dominant ones, dominant traits, alleles or traits, and the lower one are recessive. So round is dominant. So the convention of how we label them is round, the R is taken and put it as capital. For the wrinkled, we would use the small r. So the dominant first alphabet, the trait name would have been taken as the first R. Say in the color of the cotyledons, it would be yellow and green. As yellow is dominant, we would have put Y capital, and for the green one, we would use small y. Similarly here, white and small w. This way this is the convention, and just simply to say is some alleles are dominant over other alleles of the same gene, and those that are dominant, which means the exhibition of the phenotype, even presence of one copy, is called as dominant. So the law of segregation is simple in the sense that we will take a parent P1 and another parent P2. One of it is homozygous for capital B, that is blue color petals, and the second one is recessive to our white color petals. Say if we had it, these are homozygous, and if we perform, and we can also take a different scenario just for the sake of clarity. We will also take a different plants, P3 and P4. Assume that they are heterozygous. So if you see the genotype, they are heterozygous, and their color would be, the phenotype would be that blue. Both would have looked blue. Let's go with this one, the first situation, and during gametogenesis it would form something like this. And in this situation, here one capital B, one small b. Similarly here, one capital B and one small b. Let's look at the Punnett square. This is called as a Punnett square, where we can put things this way. We will just draw lines, and we are going to put the gametes here. Say the gamete here is capital B from this parent and small b, and from this parent we'll put capital B and small b. And now if fertilization were to occur between these two, we would have capital B, capital B. If it occurred between this one and this one, then we would have capital B and small b. Similarly here, capital B and small b, and here we would have small b and small b. If you look at the genotypes now, the phenotypes, this would be blue in color. This is blue because blue is dominant, and this is blue, and whereas this one would be white. So the phenotypic ratio is that we have 3:1, 3 blue and 1 white. If you look at the genotypic ratio, that would be we have one baby is to two heterozygous, capital B, small b, and I'll just move to I'll write it somewhere else. So the genotypic ratio here is capital B, let's put them up here, and capital B, and small b. We have one capital B, and then we have two of these heterozygous, that is 2, and here we have one recessive phenotype. 1:2:1. That is the genotypic ratio. It doesn't mean that it has to be always this way. It depends on the parent genotype. Here we have chosen, in this particular question, it is heterozygous. What if it was like this? P1 is homozygous for capital B and P2 is homozygous for capital small b. Then this is how the alleles would have been. So we should always remember consider the genotype of the parents. Here we would have all of them would be blue in color. Genotype is heterozygous and their phenotype is blue, all of them. So basically it is the result, or the ratios that will come by will depend on the genotypes that are present here. Let's do some problems so that we will better understand these things. Before that, the molecular basis would be simple in the sense that what you mean by segregation here is that, say for example, there is a cell and we say n is the number of sets of chromosomes, and we would say 2n because we have diploid sets, and assume that there is a capital B here and there is small b, like in plant P3. And for P4 we had similarly. And during gametogenesis they would have formed two cells, right, the gametes, and gametes are n, that is haploid. So one of it would get this way and the other one would be small b, something like this. So the point of segregation here is we have two alleles, capital B and small b. Are they going together to one of the cells, or are they segregating to two different cells is the point of it. When we are talking about law of segregation, it says that these two alleles, they segregate into two different gametes like this. And what is important about, what's smart about Mendel, is he looked at the progeny and was able to calculate using the complex mathematics. He was able to calculate or predict what is the genotype of these things. Okay, he had to do a lot of inbreeding to develop these homozygous plants first, and from there he has to do these experiments and many other. That is how one can assume how long it would have taken, because the plant has to grow, the plant has to flower, and then seeds come and then regenerate, germinate, and then form new plants. That's a lot of work. So we are going to do two problems, simple ones. Here, a breeder across two plants. P1 is tall, and P1 is tall while P2 is short. This is short. Assume tallness is dominant, so we are putting it as capital T. And because we said P2 is short, which would mean that the genotype of it is small t. In F1 generation, 50% were tall and the rest were short. Based on these observations, identify the genotype of P1 and P2. The genotype of P2 is actually implied in the question itself. They say P2 is short and they say tallness is the dominant one, which means P2 does not have a capital T which means that it has two small t's. So that is clear. We have to now discuss, it says tall. P1 is tall, so there are two chances. It could be homozygous for tall or heterozygous for tallness. So what we can do here is P1 based on the observations. 50% were tall and 50% were short. So two of them are short, right? Then, and we know one of the P2 is short because of it has small t. Those are the gametes. And here we have to, this is for P2, for P1 we have to write. So we know that one of it is, because if it is giving rise to short plants, that means it must be having its small t. And second, yeah, I'll just do a small correction here. I should put yeah. So if I had a, one second, yes, let's put it this way. Now I have two short, so one of it is that way, and this will give here. I can put all of them like this, right? Because P2 has small t, so I can put 4 t's in each of these cells. And then for P1, I need to get 2, because its answer says 50% tall and 50% were short. 2 of them should be short, and that means the P1 should have been had t here. And the 50% were tall, and it cannot be small t, because otherwise they will have, all would have been shorter. Instead it should have had capital T. Only then we will have these two will be tall and these two would be short. 50% tall and 50% short. I hope you understood the question and the solution as well. Similar questions can be made by yourselves and try to predict what would have happened in terms of predict the genotype. Thank you very much. Have fun.